Lonza Bioscience Solutions is part of Lonza's Pharma and Biotech segment. We provide life science researchers with the tools they need to develop and test therapeutics, from basic research to final product release. We are committed to supporting scientists by providing high-quality hepatic cells and expert technical instructions on the use of our products. Please read through the primary hepatocytes thawing and plating protocol in its entirety before using our cryopreserved cells. Also, be sure to prepare your biological safety cabinet for thawing and plating. Let's get started. For this protocol, you will need the following reagents and supplies. Thaw. Prepare a 37 degrees Celsius water bath. Warm the appropriate media for your application in the water bath. Once warm, disinfect the conical tube containing the medium and transfer to your biological safety cabinet. Quickly remove the cryopreserved hepatocytes from their storage location. In the water bath, vertically submerge as much of the vial as possible, up to the cap. It is important to make sure that the cap of the vial stays above the waterline to prevent possible contamination. Thaw the vial for 90 to 120 seconds. The vial will thaw from the outside to the inside, forming a spindle. Ideally, a thin ice spindle should remain once thawing is completed. Disinfect the vial and transfer it to the biological safety cabinet. Prepare thawing medium by popping any bubbles that may have formed in the liquid. To ensure that all of the hepatocytes are transferred, pipette 1 milliliter of thawing medium back into the original vial and pour the remaining cells back into the 50 milliliter tube of thawing medium. Suspend the cells by carefully rocking the 50 milliliter tube by hand for a few seconds. Centrifuge the conical tube at room temperature, following the guidelines found in the protocol. Remove the tube from the centrifuge, disinfect, and transfer to the biological safety cabinet. Pour supernatant into a waste bottle or aspirate away. Resuspend the cells by adding the appropriate medium outlined in the associated protocol. Cell counting. Use the trypan blue exclusion method to calculate the viability and yield of your hepatocytes. Due to the dual nuclei found in many species of hepatocytes, manual counting is preferred and will provide the most accurate viability and yield results. Plating hepatocytes. Use the formulas found in the protocol to determine the volume of plating medium to add to your current cell stock to achieve your desired cell density. Use the formulas found in the protocol to determine the volume of plating medium to add to your current cell stock. Using a 1 milliliter pipetter, transfer hepatocytes to a collagen-coated multi-well plate. Refer to the protocol for the correct volume of cell stock to add to each well, and for an estimate of cells per well. Place the plate in a 37 degrees Celsius carbon dioxide incubator. To ensure that cells are evenly dispersed, move plates in a north-south, east-west motion. Repeat this shaking motion at 15, 30, 45, and 60 minutes post-seeding. For 96 well plates, fill wells with 50 microliters of medium. Then add 50 microliters of cells. Remove the plates from the incubator and carefully aspirate the medium. Replace with fresh plating medium using the volumes indicated in the protocol. Post-seeding, incubate the cells for a minimum of 4 to 6 hours. At this point, the procedure differs for plates with an overlay or plates without an overlay. If you're not using an overlay, replace the medium with warm maintenance medium or application-specific medium. If you are using an overlay, cool maintenance medium on ice. Use the protocol to determine the volume of maintenance medium needed to feed your plate. Find the protein concentration of the overlay matrix on its specification sheet. Use the formula in the protocol to determine how much overlay matrix to add to the maintenance medium. Add the calculated amount of overlay matrix to the chilled maintenance medium. Remove the plates from the incubator and aspirate plating media from each well. Replace with cold overlay solution, following the volume per well guidelines in the protocol. Return the plate to the incubator and incubate for at least 2 hours before use. Replace maintenance medium daily with volumes found in the protocol. Need help? Contact our scientific support team.